Welcome to your first hands-on experience with comparative genomics. Today we'll walk through a practical workflow using the BRCA1 gene as our example. To begin, open the NCBI Gene database in your browser. This platform is a comprehensive resource for gene-specific information. In the search bar, type BRCA1 and hit enter. You'll see a list of results. Select the human BRCA1 gene entry. This step sets the foundation for our gene discovery project, giving you access to a wealth of genetic data. Let's move on to exploring what this gene entry can reveal about BRCA1. Now that you've found the BRCA1 gene entry, take a moment to explore its details. Start by noting its chromosome location, which tells you where this gene sits within the human genome. Next, read the gene summary to understand its biological role and significance, especially in relation to health and disease. Scroll further to access the sequence information. This is the actual genetic code that defines BRCA1. Understanding these sections helps you appreciate the complexity and importance of genetic research. Once you're familiar with these details, you're ready to download the gene sequence for further analysis. With the BRCA1 sequence downloaded in FASTA format, you're set for the next phase. Open the BLAST tool, an essential resource for comparing genetic sequences. Paste the BRCA1 sequence into the BLAST search box. Choose the nucleotide alignment search to compare your sequence against a vast database of genetic information. Run the search and observe the results. You'll see similar sequences from various species, highlighting evolutionary relationships and functional similarities. This process is a cornerstone of comparative genomics, allowing you to discover how genes are conserved or differ across life forms. Congratulations, you've completed your first real comparative genomics workflow.